Number one is describe the concepts of fidelity and discrimination, e.g. as discussed by Russell and Birch and others. This is about models. We use laboratory animals as models of humans. But what is meant by a model? How like the thing being modelled does the model have to be? According to the American philosopher Mark Swatowski, a model is a representation. We use them frequently in everyday life. A map, for example, is a representation of a topography, and we use maps to get from place to place, although the map is very unlike the actual topography. So a model can be quite unlike the target in most ways, but it has to be like the target that we're modelling in one or two ways, which makes it useful. So laboratory animals as models of humans or other species, how like a human does the model have to be? Russell and Birch considered that two dimensions needed to be taken into account. One they call the fidelity of the model. That is how like the thing being modelled the model has to be. And the other is the discrimination, the ability of the model to discriminate between treated and control groups. This shows some examples. A high fidelity model of humans, the highest available would be a non-human primate, because it's like humans, as close to humans as we can get when using a laboratory animal. In contrast, a Cindy doll is quite unlike humans, and so it would be a very low fidelity model. In general, intuitively, we feel we'd like to use high fidelity models, but in fact, sometimes we can use a low fidelity model if it has the ability to discriminate. For example, some people consider that an outbred rat is more like humans than an inbred rat, because humans are genetically variable. But if an inbred rat can identify the difference between treated and controls better, in other words, discriminate better than the outbred rat, then surely we should use it. Consider the question of a toxicologist who's developed a new compound and they want to know whether it's likely to cause cancer. They could put it in a rat, and this would be an expensive experiment which would take several months and would be very expensive. In contrast, they could put it in an in vitro model, a cell mutation assay, and if it causes mutations in the cell mutation assay, then it's likely to be a carcinogen, and that would be a very much cheaper and quicker way of testing it. The best example of all probably is the home pregnancy testing kit. Russell and Birch discuss at that time the available methods of determining pregnancy, which included the mouse uterus assay, the rabbit uterus assay, or the frog ovulation test, all high fidelity models and quite expensive and difficult to use. In contrast, now we have the home pregnancy testing kit, which is hardly recognizable as a model of a part of a female reproductive system, but in many ways that's what it is. It's not alive, it's quite unlike a female, but it responds in the same way when tested with a sample of urine from a pregnant or non-pregnant woman.